Welcome everybody on this sixth seminar from the SOLAS Seminar series that is on the topic of halogen chemistry from the ocean to the stratosphere. And it's actually a special seminar because it's a joint one with the IGAC Early Career Scientific Committee. And Simone is going to say something more about that because we had somebody else, Sebadies, who was going to introduce that, but he has some issues to connect. So we'll have Simone kick off with that, if that's okay, Simone.
Sure, I will try and say some words if you share the slides and then...
Yeah, I'll share the slides. I'll just...
I'm not entirely sure I know everything Seb had planned to say, but...
Yeah. I'm sure you'll find something to say. I'll just share the right one. Okay. Okay. I hope you can see that.
Yes. So this is all of our committee members from all over the world, where you can see Max and Emily are co-chairs, and then everybody else. We're a fairly new committee, started in February this year. I think we can go to the next slide. Yeah, and just like SOLAS, we're a subdivision of ICAC-GP. So in principle, our committee is called ICAC-GP IGAC. So it's a combination, not just IGAC. Next slide. January. Okay. We have 22 members, and you can see on this map which regions we represent. This both shows where we're from and where we're based. So fairly spread out, but not everywhere. So, well, I'm very Europe heavy at the moment, but hopefully we can start spreading out to more parts of the world over the next few years. Next slide, please. So our committee is at this point spread into 4 working groups. So we have communication, where Khan is the leader. He's at EPL, I think. So he's in London. And then I'm leading the engagement group, where we're working on early career researchers conference that will be virtual, and that will be on the next slide. So I'll talk about that a bit later. Martin is in charge of personal development, where we're planning to do some personal development workshops, which will also we'll start spreading the words when we have those organized. And then Bert is in charge of creating our new website that we're still working on. The goals of our committee is to, as you can see, empower ECRs to take charge of their careers, promote access of underrepresented regions, and build connections, both with IGAC, but other organizations, which is what this seminar is part of, that we're trying to connect with SOLAS and other early career committees. Next slide. As mentioned, we're planning this online conference in November. It's going to be free registration. It's going to be for early career researchers, but everybody can sign up and come and listen to the talks. We're going to have it run in series. So we're going to have it 3 times over the different time zones. And we're going to have these, we're still in the very early stages of planning it, but we'll have an icebreaker and highlight invited speakers, and post a session so everybody can present their research, and some of these personal development workshops that I mentioned, and then a lot of interactive activities so people can connect across research. Next slide. Yeah, so if you want to know more, if you have any comments, anything you want to help out with, please reach out to either Emily or Max. You can see their email addresses at the bottom. And they should, I think you can also find them on the website if that's easier. So, and they're on the IGAC website for now. I think that's it. Liselotte, I will give it back to you. You can...
Okay. Yeah. And so just to maybe complement a little bit, IGAC, for those who don't know, is very much focused on atmospheric chemistry.
Yes.
And this is where the complement with the SOLAS is nicely coming in. I'll try to introduce, and now I'll introduce SOLAS, which is also now a early career committee since not very long. And I'll try to talk a bit more about that. So for those who don't know SOLAS, so SOLAS stands for Surface Ocean Lower Atmosphere Studies. So we're looking really at the interface between the ocean and the atmosphere. The mission of SOLAS is to achieve quantitative understanding of the key biogeochemical physical interactions and feedbacks between the ocean and the atmosphere, and of how this coupled system affects and is affected by climate and global change. So it's really complementary with the IGAC missions. SOLAS has a scientific steering committee, which is comprised of 70 members from 15 different countries, as you can see here. They're a bit from all over the globe. They are changing quite often. Not quite often, regularly. That's 2 new members in the last month. SOLAS is run by a dual international project office, IPO. We have one IPO that was based in Galway, at the University of Galway in Ireland, and then one international project office in Xiamen in China at the State Key Laboratory, which is that's Lili and Shengsheng. And so there is one missing at the moment, and they're recruiting, I think, the next project officer for Ireland. They also run a master's program. That's a 1-year program on ocean, atmosphere, and climate. That is at the University of Galway, based at the University of Galway. There's a second round of admissions, which will have place in September 2023. And the courses are in Galway, but with SOLAS scientists worldwide. And then since this year, we also have an early career scientist committee, which I'm a member of, and other people here, like Lei, are also a member. As you can see, we're also coming from all the parts of the world, but a bit of an accent in Europe and Northern America. We are a strategic and operational body to coordinate and champion ECS activities. And we so far organized a workshop in April that was held online to define a little bit more what our priorities should be. Then we organized an auxiliary event at also in 2023. There will be a FLARE, a Fire Science Learning Across the Earth System workshop that will take place from 18 to 21 September in 2023. We also plan to have a side event at the WCRP Open Science Conference in Kigali in October, next October. And there is a session at AGU, which is proposed by a young early career scientist from the committee. So the session is called 20 Years of Progress and Developments in Ocean Atmosphere Science. So I encourage you all to submit to that session if you're doing research that is related to this. And then we also try to target the next call of the UN Ocean Decade endorsement application on aerosol exchange with marine ecosystems. So it's we have some things on the agenda, but that's good. It means we're active. SOLAS is also organized in national and regional networks that are coordinated by 36 representatives and 1 regional panel. And they provide annual reports of the networks. And if you're interested, you can find all the reports through the link. And as you can see, there's quite a good representation. We missed some areas are quite blank. So if you feel like you want to contribute to this, please get in touch. SOLAS, so the SOLAS science is organized in core teams. There are 5 core teams, as you can see here on the cartoon. There's one, I'll detail them quite quickly afterwards. So the first one is greenhouse gases and the oceans, which is all about greenhouse gases, as the title says it, which is not what we're going to focus on today. There's another one focusing on Andersen phase and fluxes of mass and energy. There might be some of that in here today, but not the main point. We'll have atmospheric, there is a core team on atmospheric deposition and ocean biogeochemistry, with, for instance, deposits from fire, as an example. There is interconnections between aerosols, clouds, and marine ecosystems, which we might touch upon today a little bit, but the core will be the 5th core team today, ocean biogeochemical controls on atmospheric chemistry. And as you can see here, there's a lot of halogens in there. SOLAS also has cross-cutting teams. These are integrated topics such as upwelling zones, polar oceans, or Indian oceans that span the 5 core teams, or they evaluate or they can be evaluation of the environmental efficiency and impacts of climate interventions, for instance. And they also have an accent on science and society, impacts of ship plumes, emissions on the ocean biogeochemistry, for instance, blue carbon, open ocean stewardship, and these things. So these are the cross-cutting teams that go across all the core teams. SOLAS also endorses some activities, and here you can see in light blue are the endorsed projects. So if you have a science project that is related to SOLAS science, you can ask for an official endorsement from SOLAS. They can send you an official endorsement letter, which might help to get your project funded, for instance. They also sponsor projects. These are the ones that are in dark blue. And they have some endorsed time series stations that are in green here. For instance, Pepsi is jointly sponsored by IGAC and SOLAS, for instance. Pepsi is a biogeochemical exchange, the P I forgot, processes at CI's interfaces. That's it. And oh no, it's Catch that is jointly sponsored by SOLAS and IGAC, sorry, not Pepsi. And then there's some upcoming events from SOLAS. I've already mentioned the FLARE workshop, which is organized by the ECS committee. There's a scoping workshop on SOLAS's future, because we have a next decadal plan to work on. There's a side event at the WRCP Open Science Conference, as I've already mentioned, in Kigali next October. A Goodors Summer School that is sponsored by SOLAS. Then there is an International Digital Twins of the Ocean Summit in Xiamen in China next November, also co-sponsored. The AGU session I already mentioned, and then there's going to be a SOLAS Science and Society workshop in Helsinki, and the SOLAS Open Science Conference next year in Goa in India. So these are all events, but as you can see, it's a very dynamic community, and if you want to keep up with everything that's happening, I advise you to subscribe to the mailing list, for instance, or follow us on Twitter. Okay. And I hope that gives you sort of a flavor of what SOLAS is and can do for you, or what you can do for SOLAS. And then I suggest we move on back onto our SOLAS seminar. I don't know if you can still see my screen. Yes. Okay. So today we have 3 talks on halogen chemistry from the ocean to the stratosphere. The first speaker will be Leanne Powers from the State University, then Ryan Pound, and then Rafael Fernandez from National University of Cuyo. And just before we start, I just wanted to introduce very, very briefly why we talk about halogen chemistry today. That's because actually SOLAS, the IGAC Arctic Career Committee and the SOLAS Arctic Career committee wanted to organize a joint seminar, and we were thinking which subject could be speaking to both communities. And then quickly we came on the topic of halogens, because we all worked a little bit on that. And indeed, if you look at this schematic, it sort of sums up all the interactions between halogens in the ocean and halogens in the atmosphere. And this is a figure that comes from a special issue for SOLAS Science that will appear in Elementa, the Science of the Anthropocene. It's currently in press. And so this is a great way to feature this sort of sum up of this review of the past 10 years on halogen and biogeochemical controls of the atmospheric chemistry. And so, as you can see, they're very interconnected. And for people who are not really familiar with halogen chemistry, either in the ocean or in the atmosphere, some very basic numbers. They're tiny concentrations, at least for the most reactive halogens. Iodide is nanomolar, so 95 nanomolar average in the surface ocean. Bromide, 860 micromolar in the surface ocean, and chloride around 560 millimolar. And then in the air, we have some ozone, very small concentrations, also open oceans, and some sea salt aerosols less than 10 cubic micrometer per cubic centimeter. So these tiny concentrations is what we'll be talking about, and let's see if they actually can have an influence on the global scale. And I think hopefully our three speakers will give us some enlightenment on that. And I'll give the word to Lei to introduce our next speaker. Our first speaker, that is Leon Powers.
Can everybody hear me okay? Give me a nod if you can.
Yeah. Okay,
great. So hi everybody. My name is Lei Xue. I'm currently a postdoc at SUNY ESF. I'm honored to get to introduce our first distinguished speaker, Dr. Leon Powers. Leon graduated with a PhD from University of Georgia, where she studied the marine photochemistry of carbon dioxide, CO2, carbon monoxide, peroxide, and hydrogen peroxide. Then Leon did her first postdoc in Skidway of Institution of Oceanography, where she developed this cool method to for the study of DRC photo production. Then he moved to Chesapeake Lab for her second postdoc, where she studied the algae-derived dissolved organic matter. Then they moved further northwest, and she joined the SUNY ESF in 2021 as an assistant professor. Let's welcome in for her talk.
Thank you so much, Lei, and thank you all for having me today. I'm really excited to share some of that work that was mostly done during that last postdoc. And we weren't looking for halogens in the first place, but we were working on desalination and looking at organic halogens and desalination wastewater, and we sort of transitioned into the ocean. So this is new to me too, and I'm hoping after the talk today and the discussions, I'll learn a lot more about it from all of you based on these kind of preliminary data that I'm going to show today. So let me just gear up the talk. Can everyone see this okay? Alrighty. So as you can see from the title, we'll be investigating the sources and distribution of non-volatile halogenated organic matter in the ocean and why we should care about that. And I don't really think we're going to get at the sources at all today, but that's where I think you all will come in and help me learn a little bit more about it. Let's see. So why I would be interested in halogenated organic matter in the first place is because I really want to stop thinking about carbon in terms of its importance in biogeochemical cycling. As you know, carbon is exchanged through all these reservoirs on Earth. And of course, we have put a lot of carbon into the atmosphere through anthropogenic changes, and it's a massive amount of carbon every year that is going into the atmosphere. I don't think I have to tell anyone in the audience this. So just to note, it's on the order of all of marine biomass is how much carbon dioxide is going into the atmosphere every year. And I really appreciate the oceans because they act as a major atmospheric CO2 sink. They're able to take up perhaps about half of those emissions every year. And as you've all learned in probably many classes in the past, that is via the solubility pump and the biological carbon pump, which I think is maybe way less understood as to how important or not that is in actually storing carbon. So of course you have primary production and the fixation of CO2 in the euphotic zone. And then we're transforming that CO2 into organic matter in the form of particulate organic matter and dissolved organic matter. And so some of this organic matter might be transported to the deep ocean or eventually the sediments where perhaps it can be stored for thousands of years. And so if we're thinking about climate change, that's really perhaps related to the capacity of storing this carbon in organic matter and how efficient this biological carbon pump really is. And so for our current understanding about organic matter and particularly organic carbon in the oceans is that in the dissolved pool is really a massive reservoir of carbon. So over 100 times more carbon is stored in this dissolved organic matter pool than all of marine biomass combined. And it's also comparable to that atmospheric reservoir of CO2. And what we also know is that our dissolved organic matter is very interesting because it's a dynamic component. There's many, many sources and sinks, but it's really hard to study because it is a variety. It's a very heterogeneous mix of compounds that we don't really know the composition of. And so we know there has to be a very large range in the reactivities of all these different compounds. And of course, I always like to tell students that it's really important to study because there's an entire textbook just on the subject of the biogeochemistry of marine dissolved organic matter. And so if we're thinking again about that biological carbon pump, there's sort of a conundrum here in my mind because we have that labile DOC in the surface ocean that is coming mostly from primary production. It gets either degraded maybe photochemically or microbially or just reworked, and only a very small portion of that makes it down to the deep ocean. But once in the deep ocean, perhaps it can be stored for thousands of years. So if it's so reactive, why is it being stored for so long? And so we know based on radiocarbon data, this is from one of the seminal papers from Bauer, Williams, and Druffel in 1992, we know radiocarbon ages of organic matter in the ocean. In the surface ocean, it's much younger. And then in the Pacific and the Atlantic, there are differences as well. So in the deep ocean in the North Atlantic, the apparent age of organic carbon is 4,000 years old, and that in the North Pacific is 6,000 years old. And perhaps the rates of respiration in the deep ocean are very slow, and this is reflecting a difference in transit times between the two ocean basins. And I'd like to go back in time even a little bit further and thinking about even our understanding of organic matter in the ocean back in the 60s. And you can just see from the title of this paper, dissolved organic carbon from deep waters resists microbial oxidation, which means it's probably really difficult to degrade once it's in the deep ocean. And so for a long time, we kind of, I think, thought about this organic matter being very refractory, a pool of carbon and associated elements that's just uniformly distributed through the deep ocean. And so over the years, we've gathered a lot more data, and the general consensus is in the surface ocean, you have these higher concentrations of organic carbon, and that organic carbon is younger. And once you're in the deep ocean, concentrations are lower, and it's a lot older. And so this can be described pretty well just by a 2-component model based on DOC concentrations, the delta 14C of DOC and the delta 14C of the DIC. And we kind of think of this refractory organic carbon as being uniformly distributed with depth, with that labile DOC in the surface ocean overlaying it. We even have what I think is interesting from some reevaluations or more recent measurements from 2009 of the radiocarbon in the deep North Atlantic again, and we still have that radiocarbon age of 6,400 years. But what I thought was really interesting in this paper is that they did Keeling plots that may suggest that a good portion of that DOC might be much older. So based on these plots, you might predict that a good portion might be almost 60,000 years old. And also, we've been also thinking for a long time that this is not just the age of everything. You know, it's more realistic to think of it as a continuum of materials that range from modern to basically radiocarbon dead. And so just as a review for everybody, I think it's always nice to show how this radiocarbon decay might really skew what we're thinking about in these apparent ages. And so we know the half-life of radiocarbon is about 6,000 years old. So that means by the time you've gone through 10 half-lives, you're essentially radiocarbon dead. And if you do just a simple mass balance calculation, I don't even know if this calculation is correct, but even if you only 10% of your organic carbon in the deep ocean was 60,000 years old, the rest could be modern, and you would still get that average age of 6,000 years. If you did a simple residence time calculation based on the estimated flux of DOC from the surface ocean to the deep ocean and the amount in our entire reservoir, we actually would have a residence time of about 400 years instead of 600 years. So all of this background on carbon is just to reiterate the point that it might be time to start thinking about other elements within DOM to try to start understanding its reactivity. And that really leaves us too in thinking about all of the sources and sinks of organic matter and just how variable they are and how poorly we understand, I think, at least marine dissolved organic matter reactivity. I highlighted atmospheric deposition and sea to air exchange just for the group here today. But again, I think it's time to stop thinking about it as this refractory pool that's just uniformly distributed throughout our deep sea. And so this is where halogens are coming in. And so I first really got interested in brominated organic matter and halogens in general because we were working with sargassum and we were doing these incubation experiments, and we were just trying to see how much organic carbon sargassum released over time, both outdoors exposed to sunlight and indoors exposed to just visible light. And we found that over time, they release a lot of organic matter as dissolved organic carbon. And we also did some high resolution mass spectrometry to characterize the organic matter, which is shown here in this figure, in this elemental diagram. And so you can see we are plotted ions that contain just carbon, hydrogen, and oxygen. And then we found ions that contained carbon, hydrogen, oxygen, and bromine. So it's well known that algae, these brown algae, contain brominated phenols. They can definitely release halocarbons into the surrounding waters. I think that's been well studied too. So it's no surprise we found these, but we had never really seen it before in looking at bulk organic matter from seawater. And so we plotted our concentrations of brominated DOM from sargassum versus DOC, and we had quite a high ratio of carbon to bromine in these samples, almost on the order of what has been reported from the ratio of carbon to sulfur. So it indicates that, to us at least, that maybe halogenated organic matter is a significant pool of halogens in the ocean, and also maybe sargassum is a source of halogenated DOM, at least in the Atlantic Ocean. And so in starting to think about this, it turns out that halogenated DOM has really, I think, been ignored by at least people in the DOM world for a long time, and I'm not really sure why. I mean, one guess might be that halogenated organic compounds don't have the best reputation, as we might talk about a bit here throughout the rest of our talks today. But what I think has been really cool about all this research about anthropogenic halogenated compounds is that it has led to the discovery of thousands of naturally occurring halogenated organic compounds. And so there's a lot of reasons why we should care, highlighted in this nice diagram from this 2017 paper. But we know that marine organisms can produce a wide array of halogenated metabolites. And maybe this is a defense strategy. So another big field of research in natural products is looking for bioactive organic halogenated compounds. And so clearly we know a lot of organisms can do this. But my interest is really that this has to be related somehow to the biological and microbial carbon pumps, and therefore has to be related to DOM cycling in the ocean. And we know that halogens can be incorporated into the DOM pool via these enzymatic and abiotic pathways. And so since that is the case, we know then that these halogenated organic compounds can then exchange with the atmosphere. And it should be this interesting cycle or big component of this cycle in itself. And in general, as I mentioned, that DOM reactivity is poorly understood. And so maybe studying halogen organic matter might be a way to better understand marine DOM reactivity in general. And so coming from a photochemical background, one thing I've been thinking about for a while is how do you make halides reactive? And of course, we know that reactions with hydroxyl radical or triplicate sided state DOM can form these reactive halogen species, and they can go on and react with DOM to form halogenated organic compounds. But I think, biology is probably ruling the world here too, so I can't ignore, I need to start appreciating these enzymatic pathways as well in terms of these halogenated organic compound formations. So I think we're going to hear more about the volatile organic halogens later on. So again, I'm going to try to focus on these non-volatile species. And so in thinking about what the distribution might be, well, we found some in our sargassum exudates, and there's been a few other studies that have found either iodine-containing metabolites from Prochlorococcus or iodine and chlorine-containing metabolites from E. huxley. And so there's got to be a way that we can analyze total organic halogens in the ocean. And then I just wanted to say too that we've known about it for a really long time, or at least the evidence for it. There's a really nice paper from Truesdale in 1975 that discusses an unreactive iodine component and suggests it has to be organically bound. But of course, the analysis of organic halogens is probably going to be very different because we have a low concentration of organic halogens over this very high highlight background in seawater. And so just to get around this issue, we adopted a rather simple method that basically is utilizing solid phase extraction that's going to concentrate organic matter, and then a halide cleanup step that is going to remove any residual salts that is reported in this paper by Mendez and Diaz. And so basically, we've taken data from only a few locations. One is a coastal cruise in the Mid-Atlantic Bight off the east coast of the United States, and also from two long-term monitoring stations, the Bermuda Atlantic Time Series Station in the North Atlantic Gyre and the Hawaii Ocean Time Series Station in the North Pacific Gyre. And so very limited data again, but I think what we'll see now is just that the differences in these concentrations is pretty interesting, especially if we're thinking in terms of carbon. So here we go, following our steps from Mendez-Diaz et al. We're simply filtering a lot of seawater on our cruises, and then we're doing the solid phase extraction. So we're using two types. One is a reverse phase to get these more non-polar compounds extracted, and then followed by a weak anion exchange to get even more polar compounds that are missed by that PPL resin. Then once we're back in our lab, we are going to reconstitute these extracts in water. These are methanolic extracts. So basically they're dried down and redissolved in pure water. And then we use silver cartridges to simply remove residual halides. And then we analyze the concentrations of organic halogens by ICP-MS-MX. And this is giving us our organic bromine and organic iodide, and even organic chlorine, even though the detection limit's a little bit higher for organic chlorine. And so first, in the Mid-Atlantic Bight, we can see a couple trends that you might expect from being in coastal waters. For one, we see differences from low salinity to high salinity, where our organic bromine concentrations seem to decrease as you go to higher salinities, and organic iodine seems to increase. And then for some of these offshore stations as well, we have lower concentrations as we go into deeper waters. And it seems to be that they're almost relatively stable with depth for both the brominated and iodinated organic matter. And so in thinking about why this might be, well, for one, maybe we can look at the ratio between bromine and iodine in some of these surface samples. And so when we do that, we have somewhat higher bromine to iodine ratios and maybe these more productive waters, or waters that are at lower salinities. And then as we get offshore, that ratio seems to be maybe stable with depth. But of course, these are, all of these samples are influenced from the coast. So of course, we should move offshore into the middle of the gyre. And so when we do that and compare to our stations near Bermuda, you can see, just like you would expect from going to coastal offshore waters, we have much lower concentrations of both organic bromine and organic iodine. And also what's interesting, I think, is the changes in these ratios between bromine and iodine. So maybe organic iodine is preferentially removed versus organic bromine, but again, very speculative based on these preliminary profiles. And so the next thing to do is to look at the distribution between our two ocean basins, right? And so I was only able to get organic chlorine concentrations at our BAT station, and they're quite noisy. But one big takeaway is that it seems like perhaps that's much larger concentrations than the brominated or iodinated DOM. And what I will say too is we also looked at bromine and iodine in those more polar wax extractable organic matter fractions. And so they follow somewhat of the same trends, but there are some interesting differences. So I know it's just a lot to look at these profiles in the slides. So the big takeaways here is, one, does the chlorinated DOM actually decrease with depth, or is just this data too noisy to actually pull that out? And is it truly much higher in concentration than both the brominated and iodinated DOM? Another question is, do BR and IDOM decrease with depth until about 1,000 meters, and then is it relatively stable after that? And then, as you can see, we have significantly different concentrations at depth between our Pacific samples at hot and our Atlantic samples in Bermuda, which was surprising to me because we first found those brominated organic ions in Sargassum, which would be in the Atlantic. So it was really surprising to me to find much higher concentrations of brominated DOM at depth at hot versus at BATS. And just to point out with our extractable carbon that these trends are different from organic carbon. So a really interesting way to think about maybe the differences in reactivity between these ocean basins. And so I know I'm probably getting close to time here. So for the last part of this talk, I thought it would be fun to take information from two profiles and apply it to the entire ocean, like any good oceanographer. So we can take our total extractable organic halogens, and we can then compare them to organic carbon. So we know the efficiency of organic carbon extraction. We don't know it for the halogens, but we're going to basically use this information and the ratios between carbon and our halogens to calculate an inventory of our organic halogens in the ocean. And so we can do that based on information of our distributions of organic carbon in the ocean and how much of that could possibly be extracted by this PPL method and this wax method. And so when we do that, these are just the ratios between carbon and bromine, between our different extractions, and also between carbon and iodine. And so just to go through this quickly, we have a bunch of averages over these depth ranges, and we have a total inventory of about 0.7 petagrams of bromine in the ocean. And when we do that for iodine, we have about 0.4 petagrams of iodine. And I think looking at this 1 petagram inventory, maybe will give some interesting insights into DM activity. So working with Feng Chen, who's at the University of Maryland Institute for Marine and Environmental Technology, on one of our BATS cruises, he looked at the abundance of dehalogenase and halogenase genes, and perhaps, at least for our most abundant dehalogenase gene, they may be increasing with depth. And so is the dehalogenation of DOM coupled to its oxidation? And is that a link to the carbon cycle? Another thing to think about is when we plot them all together versus, say, our brominated DOM, is this trend real? Is there something to pull out here? I mean, this is all really preliminary data, but I think the point here is just it's worth looking more in detail at. Another thing we've talked about is that oxygenations might be involved with dehalogenation because you can replace a halogen with an OH group. You can also replace a halogen with an H. And so when you do that, they might be more readily oxidized. And so we also saw that oxygenases perhaps increase with depth. Again, this is really noisy, which is not the case perhaps for oxidases, which we don't think are going to be involved with dehalogenation. And so I'm going to skip over this because, of course, I put way too much into this talk, but we'll just get to some of these further thoughts. One being that, of course, what I've shown you here today is not a lot of data, but I think it really does hint towards the differences in DOM composition between ocean basins, and that halogens are really worth looking at in terms of organic matter. And of course, these estimates are small compared to organic carbon that you can extract, but still combined, if we're just thinking about bromine and iodine, 1 petagram is a lot of organic halogens. And so it begs the question, what are we missing by doing this extraction? How much does it make it into these extractable pools? And using a similar approach, they've reported in this paper that the organic sulfur inventory is about 6.7 petagrams. And so perhaps halogenated organic carbon, or sorry, halogenated organic matter is of comparable size. And so another big point is that we really haven't gotten a good method yet for organic chlorine. So we're still going to work on that. And once we do that, perhaps we can even more robustly say what this inventory might really be. And so for one, I would love to establish a more robust inventory of halogenated organic matter in our world's ocean and discover if any of these compounds can be used as tracers or just in a way to understand these different processes better and DOM reactivity in general. And so will answering any of these questions help us better understand global carbon cycling? I really hope so. And I'm also hoping that you all will give me some ideas about that as well. So that is all I have for this talk, but I'm happy to take any questions and looking forward to the discussion.
Any question for Dr. Powers? Thanks for this really insightful talk, by the way.
I always try to put too much into a talk, so sorry about that.
Yeah.
I see one from Lucy.
Okay. Yeah.
Yes, hi, that was a lovely talk. Thank you. I don't know what I can see now. I'm just going to see. Yes. I was going to ask Leanne a couple of things. So well, one is around that particulate. Whether you think there's any sort of particulate phase halogen organic material going on. You don't pick that up in your method of I guess. You've just got the dissolved side of things.
Right. So based on the filtration, I mean, that's the only way I'm saying it's dissolved is operationally. So we haven't looked at particular organic matter, but that would be really interesting. I think if you could extract them from filters, it might be a way to do that, especially during blooms. I mean, we see some interesting things around the deep chlorophyll max, especially with iodine. So maybe that's something worth doing in the future.
Great. Yeah, my second one, in fact, we just got a paper, Liselotte will have it in her inbox from about an hour ago, on measuring iodine speciation at a coastal site, which includes sort of the organic phase from looking at the difference between total iodine and iodide and iodate and all the other bits. So the concentrations, you know, match up with what you're seeing, 20 nanomolar or something. And I was just really interested because for iodine, what you see is very much a surface maximum, right? And then decreasing, and that's quite different to the bromine. And I wonder, you know, we've been discussing what the formation mechanisms are. I guess it's a combination of, you can have the reactive iodine species, I2 and HOI, reacting with organic matter, but there's some clue in the literature that you might have iodate reacting as well and forming these sort of iodate organocomplexes. And I guess your data would say more it's the reactive iodine species because of that kind of extreme profile. So I don't know if you've got any comment on that, and particularly the differences between the iodine and bromine vertical profiles.
I know. So yeah, it's one thing I'm also thinking about in regards to your question, which maybe doesn't answer it, but also the exchange of halogens for each other. And maybe is organic iodine the most reactive, but also perhaps it exchanges with bromine or even chlorine, and maybe the other way too. I don't know. I mean, I think some early papers on it occurring with just halocarbons, right? But
yeah, sure. That's a good point. Yeah.
I'm wondering if some of these exchange reactions are explaining it, but it is interesting that it is so much higher in the bromine with depth.
Okay, thank you.
I don't see any other questions, but if I can just ask a small one. You mentioned that the fluxes are estimated, like the DOM fluxes to the to the lower ocean. How do they actually estimate this? I don't know this.
Oh gosh, good question. I should know this too. I mean, it's based on global inventories and ocean circulation, I'm pretty sure. Okay. I should definitely review my oceanography a little bit better.
Oh, sorry. That was a bad question.
But of course, you know, it's a huge error bar associated with that 1.8 petagram of carbon per year that makes it, you know, to the deep ocean. So is that real? I don't know. That's my question. I think just constraining all of these fluxes is going to be a big challenge, but really important to get after.
Yeah. It's always surprised me to hear that the DOM is so refractive, but yeah, there is lots of things going on there.
Oh yeah.
Yeah, thanks. So if there are no further questions.
There's one. David, go ahead.
Hi, Leanne. This is Dave.
Hi, Dave.
I have a quick question. I noticed there's a lot, there seems to be higher concentrations in near surface waters. And the halogen, if it's a simple iodine carbon bond or bromine carbon bond, they're probably going to be photoreactive. Have you looked at that at all?
Yeah, there's a nice chapter from 1995 with Richard Zepp and somebody else. So I can send, I think it's Zepp and Rittmiller, where they talk about dehalogenation photoreactions and how you can have maybe perhaps a direct photoreaction to cleave that bond. or also indirect reactions with reactive oxygen species, say. But I haven't seen many people looking for that, maybe because it's difficult to measure, say, the loss of organic halogens. And then also on the flip side, you can get the photoformation of halogenated organic compounds via the same reactive halogen species type reactions. So it's difficult. I did try to look from some of these desalination wastewater samples that I had to look to see if you were losing organic halogens during irradiation or forming them. And I basically really didn't see a change at all. So I don't think the methods are sensitive enough that I was using, or it is just this mix that makes it really difficult to know exactly the rates of either one.
I think maybe we should move on to the next speaker.
Great. Thank you all.
Questions?
Thank you. There's a question in the chat from Ming, but we'll keep that for the end then, Ming.
Okay. So our next talk is going to move us from the ocean to the ocean-atmosphere interface. Dr. Ryan Pound from the University of York did his PhD with Lucy Carpenter and Matt Evans on modeling of ocean deposition to the ocean, and has later started a postdoc at the University of York where he's looking at the emissions of iodine, so going the opposite way. And I think that's what he's going to focus his talk on today.
Yes, that is the plan. I will get my slides up. Here we go. Yeah, so as Simone mentioned, I'm going to be talking about ocean-atmosphere exchange and coupling the process of ozone going into the ocean surface and subsequent emissions of iodine compounds. And this will be presenting some work that I've been, some modelling work that's been the past couple of years, but also I've got some shout outs to work being done from around the rest of the group that's all sort of combining together to be this wider story of ocean atmosphere exchange of iodine. So with a quick start on the ocean side of things, this is a bit of a brief overview of iodine species in the ocean and what then is eventually emitted in terms of the inorganic side of things. And we have this at the top here, the two largest iodine compounds that we that exist in the ocean. We've got the iodate and iodide, which is where the speciation sits, and it's this iodide that reacts with ozone in the ocean surface that produces out inorganic HOI and I2 that can subsequently be emitted. Of course, the iodide being sort of the key part of this is something that we care about in terms of how it's distributed. And as Liselotte mentioned at the start, we were talking on the order of nanomolar here in terms of concentration in the ocean surface. And this is some work that was done by our group previously in York. And we can sort of see there the rough global distribution, and that we have the highest iodide sort of in tropical warmer waters, and this decreases as we go towards the poles. And I'm focusing on the inorganic side of things because if you add up all iodine emissions, both organic and inorganic, 75% of all iodine emissions is from our I2 and HOI side of things. So this is the biggest lever we can start to use to understand iodine emissions and their impacts on the atmosphere. So hopefully this is now, I'm going to try and convince you that you really should care about iodine and its effects on tropospheric photochemistry and particularly ozone. So we have all of our iodine emissions, if you run in global chemistry transport models, they decrease tropospheric ozone by about 6%. So this is quite substantial, and it's roughly comparable to the increase that you get in tropospheric ozone from isoprene emissions. And it's important because there's an order of magnitude more amount of research and papers being put out for isoprene than there is iodine. But actually, to understand tropospheric ozone, you need both of these in tandem to really sort of get that model simulating ozone as as accurately as possible. And iodine is really important for determining background ozone. So in this plot here, I've got the more purple the map looks, the more important iodine is, the larger the effect that iodine has in modulating surface ozone. And the more green it is, the more important isoprene is. And as you can see, a vast majority of the world is purple, and all of this area, although it's over the ocean, ultimately helps determine the background ozone levels, which end up running over the continents. So there's a sort of an offshoot here of, although I'm focusing on ozone, there is impacts on OH and sometimes methane lifetimes, but iodine is quite negligible in this, and there's some competing factors in chemistry as to why this ends up balancing out. So that's sort of quite a tangent, and we can talk about that if need be, but ozone is going to be the main focus here. So if you was to make a nice simple schematic of all of this ocean atmosphere exchange, you'd end up with something that looks a bit like this, where we have ozone coming into the ocean surface, reacting with iodide, and then we've got some inorganic chemistry happening in our model, because there's, as I said, we're focusing on the inorganic first as the priority, and lots of mixing between both the bulk ocean and the atmosphere, and as a result we get some halogen compounds being emitted, but the bulk of that emission comes in, as I said, from HOI and I2. Now, the first bit I'm going to focus on in this model, in this new model particularly, where we've started to sort of update individual parts of this, is with this reaction of ozone and iodide. So this is a paper published in 2018 by Moreno, and it covers this sort of the kinetics of this ozone and iodide reaction, and particularly the mechanism of which it works. And as we see here, at these very low concentrations, which are comparable to where we are in the ocean, all of this happens in the aqueous phase. But as the iodide concentration increases, this starts to become a surface reaction. And this is important because the ozone plus iodide rate coefficient that's been used in previous models uses much higher non-environmental concentrations, so sort of approaching molar concentrations of iodide. And as you can see, this has a huge effect on that uptake coefficient. So one of my colleagues at York has an experimental setup that looks something like this. We have a flow tube where we can have either a sea surface analogy flowed through it, or actual sea samples. Ozone can then be passed across the top of that, and we can measure the loss of ozone and therefore work out that uptake coefficient. And importantly, this is being done at environmentally comparable concentrations. So we should be in that scheme where we are best replicating the actual uptake of ozone in oceanic conditions. And what's the result? Well, as I said, the one that's been used previously, so that's these collection of green dots here, wasn't under environmental conditions for either ozone concentration or iodide concentration, whereas this work has been, which is the pink dots up here. You can see there's quite a difference in terms of the temperature sensitivity of that rate coefficient. And there's also a much weaker temperature dependence now, and it's more in line with some of these other non-temperature dependent studies which have been conducted in the past. What does all this mean though for our emissions? So if I run the model using the old rate and the new rate, so the old rate being the yellow line here and the new one being the blue, we can see that there's now a substantial change in our temperature response to our total iodine emissions. So we now have much higher emissions at lower temperatures, and slightly lower emissions at higher temperatures. So we've already managed to sort of have quite a substantial impact from this lever. The next sort of step on this diagram I'm going to focus on is actually what is our iodide doing in the SML. And previous work treated this iodide as a constant, and it was just fixed. So if, say, if the ocean or the area of ocean we was looking at was 100 nanomolar of iodide, the bulk water and the SML water would all be kept at 100 and be left at that. But actually, that's not necessarily the most physical way to represent this. The SML is this area of sort of intense amounts of air exchange with both the ocean and the atmosphere. And the concentration of the iodide is dependent on how efficient those mixing between all of those components are. So if we let mixing take over and we're no longer fixing the iodide concentration as a constant, which we can see here in the blue line where it's constant, if we let it mix and be controlled by that mixing, we get this sort of depletion that's happening at lower wind speeds. And at lower wind speeds, it's depleting simply because we're not mixing, there's not enough turbulence to drive the mixing of this iodide back into the SML. And the result of this depletion that happens is we get a dampening to our total iodine emissions, which is pretty interesting. So at these wind speeds, which aren't uncommon, we start to see the model is now predicting a decrease. But does this depletion of iodide actually exist? Well, there's some other work which has been done by the group at York we've got here. So we can just see the ones to pay attention to is the iodide here, which is these blue crosses on this figure, and these are from SML and underlying water samples taken on the Connect cruise in 2021, and this is all very remote ocean. And what we can see here is that the SML, the surface of the ocean, has this depletion of iodine in it. We're talking, this is sort of on the orders of 40% here, which is a much greater depletion than what my model predicts. So there's something to explore here in terms of why is this depletion greater. There's a lot of factors that my model doesn't have in it at the moment, but what we take away from this is that depletion isn't a numerical fantasy from model land. There is some form of experimental and real world backing that this is something that we should be observing. So the next step, now we've got ozone that has reacted in the ocean surface with some form of iodide, is now all of the subsequent chemistry that happens from that initial reaction. And this is, again, something we've been expanding upon. And the one aspect of this, among several that has happened that I'm then focused on, is a salinity dependence that happens in this reaction. This is some work that's been published again more recently by other research groups. But as the experiments sort of added chloride and bromide to just an iodide solution, there was a dampening of the total iodine emissions which were being observed. And this work concludes that, in fact, the rates being used in the model, specifically the equilibrium, see these two species that are circled here, were overly favouring I2Cl, which allowed HOI to be converted to I2 much more rapidly in the model than it was in the samples which are being analysed for emissions. So there's a new proposed equilibrium rate for this, and if we put that into the model, what we see is we now have the old rate and the new rate, and again, old is yellow and new is blue. We have again our total iodine emission being dampened, and we're bringing that bar further down again. So there's quite a decrease there. And this is sort of just a little sort of side note here, is that these are obviously truncated and simplified chemical pathways, but my model also does have a increase predicted when bromide as well as chloride is included in the system. So is this similar to the fact that there's some equilibriums in the chloride pathway which is too efficient, or is there something else? And this is something that we could explore with further experiments down the line. So finally, the emissions. We've worked our way across from the start here with the ozone all the way across to where we are with iodine emissions. And all of these changes along the way, what has that done to our total model predictions? Well, here we compare two sets of equations. So the work that was previously done by Lucy Carpenter, my current supervisor, is represented with the black lines here, and the work that we've done, that I've been doing along with the rest of the group, is represented in orange lines here. Now, from both of these box models which have been used, we develop a set of parameterized equations. And this is important because these parameterized equations are effectively taking all of the complexity of ocean atmosphere exchange and condensing it down into some simple a nice simple equation where you can put in your ozone concentration, your temperature, your iodide concentration, and your wind speed, and get a predicted flux out, which is very good for use in global models. Going back to the start where we're using global models to understand the importance of iodine. Now, one of the big changes, which you'll notice on the bottom right of this panel here, is that the previous equations had no temperature dependence, whereas now this work does. And you can see there that there's quite a large change over sort of typical ocean temperature ranges in our total iodine emissions compared to the previous work, which is just sort of predicting a constant here. The second biggest difference is in this top left here with the wind speed dependence. Now, the old model, as wind speeds got lower, predicted unphysically high iodine emissions, and these sort of, as wind speeds decreased further and further, iodine emissions shot off. And to sort of control this to some extent, a wind speed minimum cap was put in to say, if you want to use these equations, don't use a wind speed below a certain value. Whereas and as you can see, the orange line here from this work doesn't have the same problem as this new model, thanks in part to that limitation of iodide becoming depleted at low wind speeds. As you have less and less iodide, you have less ozone reacting, and therefore you've got reduced HOI and I2 being created and less emissions. So this system is now self-compensating and behaving in a much more physically understandable way. That's all good comparing to another model, but what does it mean in terms of some lab experiments? Well, this is comparing here from a selection of lab results of I2 flux, which is measured from either sea surface samples, artificial seawater, or just iodide solutions. And in general, we can see that the model in blue there is underestimating the experimental results in red. There's quite a lot of nuance in this, and there's a lot of different ranges of ozone and iodide that's been used, a lot of different experimental setups. But one of the key differences that we've found is that actually, if this flow tube, in case the experimental vessel where the ozone and aqueous solution are reacting is being stirred, so has a more turbulent mixing, in essence, the model is much better at predicting those experimental results than if that solution isn't being stirred. But as I said, there's a great deal of uncertainty and complications added by the experimental conditions, and this in part is going to inform future work that we do in York, is to better create, if you like, an actual ocean analogy in a piece of glassware. Although obviously there is a great deal of complexity in simulating an entire ocean inside a tube which you can fit on a lab bench. But the model isn't completely unrealistic in our most sort of representative cases, which is quite good. So, looping back to some global maps again, so that's where I feel most comfortable. If we take this model and we use that parameterization that we generate from it, what we have is some new emission numbers. So this is our total iodine emission map here. And if you cast your mind back to what iodide looked like globally, we had much higher iodide at the equator. But that doesn't necessarily translate to higher iodine emissions at the equator. In fact, there's quite a different spatial distribution, and it's not just being dominated by where our iodide is. There's now sort of a great deal of complexity here. And if you compare using the old equations from the previous work done by my supervisor Lucy to the new emissions, we now see that actually there's been quite a large spatial change in where our model is predicting these emissions to come from. We're now seeing much higher emissions from colder waters, which, as we've seen, is in part largely down to the fact that we've now got a temperature dependence in those equations, and also the change in rate constant means that this reaction is happening much faster in colder waters, as well as lots of other parameters in terms of how important the mixing is in these regions and how important that depletion might or might not be. But we see that all of this adds together, seeing now we have a 25% increase in our predicted total global iodine emissions. So where this sort of talk is ending is where I'm currently at in terms of work, and now starting to analyse, okay, looping back to the start of this talk, we've now got this new improved iodine emission parameterization, our new best guess at what total iodine emissions look like. If we feed that back into the starts, what does this do to tropospheric ozone? How much of a change does this? How much more purple does our world map look in terms of how much important iodine is in all this picture? And that's sort of where we're gonna. That's the area of this that I'm currently working on. So I've given a bit of a whistle-stop tour here for the last 2 years of my research. But if I send you home with 3 messages, is that in York we've developed this new box model which incorporates lots of advances from our group and beyond on previous work, and we use this model to predict inorganic iodine emissions, and these changes remove a lot of the previous limitations of the last iteration of this work, if you'd like. And we're now working on using this box model to inform global modeling on a global scale, and really add a greater refinement to our understanding of ozone in the troposphere from all of these ocean-atmosphere interactions. So hopefully I've not lost anyone, and you've all been interested and sold that iodine emissions are as important as I claim them to be. And I'd like to thank you all for listening. If you have any questions, I'll be happy to answer them.
Thank you, Ryan, for an interesting talk. I think there's already one question in the chat. And
there's some hands up as well.
what, Liselotte?
There are some hands up. There was Amy, Lise, and Vanessa who had hands up.
I think they were just clapping.
Oh, okay, sorry.
Yes, ask away.
Hi, Ryan. So great talk. Thank you very much. I have 2 questions. One was the wind speed threshold that you pointed at, comparing with the old scheme or the old equations that you used, was about 4 meters per second, something like that.
Yeah,
between 3 and 5.
that was about 5 meters per second. I think it is how it's, yeah. Okay.
And the other one is what's the global emissions that you get with the new expressions? I mean, in previous with the previous equation, I think it was about 2 teragrams per year of iodine. And what's the result for the new one?
Yes. So the previous work looks at it ends up being about 2 teragrams of inorganic iodine emissions. So that's HO and I2 combined. This is nice and easy in terms of round numbers, because the old work is about 2 teragrams. We get a pretty much exact 25% increase. So we're looking at about 2.5 teragrams, which would bring our total iodine emissions, including organic as well, to about 3 teragrams, I think, is okay. A new rough number off the top of my head. So there is, yeah.
Okay. Yeah, that makes sense. But then it's distributed more into the high latitudes and not that much in the tropics. You have a larger proportion in the high latitude.
Yeah, that's a change in distribution is something that we're starting to explore now. So I've in preparing this talk, I've just about managed to get the new emissions in and predicts that on a global scale. And as soon as sort of this talk's done, I can go back to my desk and start working on what does that change in distribution do. But the increase in higher latitudes is going to be quite interesting in terms of what what that does to our global ozone distributions in the troposphere.
Yeah, sure. That's great. Excellent. Thank you very much.
Thank you.
Any other questions? Bill, go ahead.
Then can I ask my question?
Yeah.
Anybody?
Go
Okay.
ahead.
All right. Great talk. I really enjoyed that and learned some stuff, obviously, because this is not my primary field. But I was just wondering if you wanted to make a comment on ocean acidification. I thought that one plot of the very significant drop in iodine emissions with pH was pretty interesting. But I don't remember the pH scale that was on the bottom of the plot. It may be that it's such a small change that it's insignificant. But have you ever thought about that?
So that pH scale is about. Hang on, I can just double check from the beginning. Yeah, so that goes from pH 6, 7, and 8. So there is a pH dependence in this reaction, and you'd expect there to also be a sort of funnel through to the emissions. It's not something that we've focused on yet, but that's definitely an area that we've had conversations about internally, at looking at in the future and seeing, using forecasts for pH changes to see if that would impact in sort of future modeling scenarios.
Thanks.
Thank you.
Any other questions? There's one in the chat, Brian, about the name of the ocean atmosphere exchange model.
Yes, so my box model that I use doesn't really have a name yet. We have been trying to work out a series of letters which we can use to give it an appropriate acronym, but there is nothing yet. We're going to publish that work soon, hopefully. The previous box model work, which was done by Lucy, who's also here, I don't think that either has a name either, but there's a whole paper describing that as well. And for the global model side of things, for we've been doing the iodine emissions and understanding how they impact ozone, we've been doing that in GeosChem, which is a global chemistry transport model and is used across the world and is open source, and there's lots of stuff online about that. So I'd be happy to point people to, for all three models that I've talked about briefly, I can point anyone to sort of more resources about them if they need to.
Great. Any last questions before we move to the final talk of the day? No? Thank you, Ryan.
Thank you very much.
Liselotte, did you have something?
No, I was just going to say thank you for the talk, and also just to point out that after the 3 talks, we can have another 10 minutes of discussion on all the talks if people have more questions by then.
Okay, our last talk is going to take us from the ocean surface to the stratosphere. So we take the next step, and that will be done by Dr. Rafael Fernandez, who has a PhD from the National University of Cordoba in Argentina, and is now a professor at the National University of Cuyo. He'll be talking about very short-lived halogenated species from the ocean and how they influence the release of reactive halogens to the troposphere and stratospheric ozone depletion. Go ahead. If everything works.
Do you hear me? Sorry.
Now we hear you, but we
I
don't
was
see your
muted.
screen.
Okay, sorry. Okay, now I go back again. There we go. Did you hear me? Hello?
Yes, we hear you. We can hear you.
Yeah, we can hear you.
Okay, thank you. Sorry. Sorry about the short delay. So now let's move into how these halogen emissions impact on the atmosphere and mostly into the stratosphere. I was thinking on talking about the tropospheric impact or the stratospheric impact, but I thought to focus on the last ones. And so this is just to highlight how the halogen study in the atmosphere kind of began. So long-lived halogens, which are species that they don't photodecompose in the troposphere, they reach the stratosphere, and they were first studied to affect the ozone layer. These were mostly CFCs, chlorofluorocarbons, and HCFCs, but also some halogens, which are equivalent compounds, but including bromine atoms. And they are unreactive in the troposphere. So they have lifetimes, which are here on the table, of about 10 to 100 years, mostly, with the exception of a few of the compounds like methyl bromide or methyl chloride that are of about 1 year. So these species, once they are emitted, in this case, these are mostly anthropogenic emissions. So they are transported all the way up to the stratosphere. And once they reach the stratosphere, because they have ultraviolet radiation, they photodecompose and release halogen atoms, which can easily destroy ozone. So after the implementation of the Montreal Protocol, the emission of these species started to decrease, but because of this really long time, it will take several years, and it's even by the end of the century where we will reach some halogen abundances, which will be equivalent to what we have before 1980. So what's the motivation to study very short-lived halogens or short-lived halogens? These species have tropospheric lifetimes, which are of below 6 months, between days. For example, for the iodine compounds, these species have lifetimes of minutes or days, and for the bromine compounds, mostly from a few weeks to a couple of months or 4 months. So when balloon and aircraft observation of the total amount of bromine in the stratosphere were performed, which are all these symbols over here for the different years, whenever we compare the total amount of halogens emitted from halogens or from methyl bromide, anthropogenic or natural contributions, you could never reach the total amount of bromine in the stratosphere. So there was a gap of bromine in the stratosphere, and that could be explained only by the inclusion of these short-lived halogen emissions, which were mainly of natural origin. So how do we translate this into a chemical or reactive transport scheme? So basically, we need to have, or we knew there were about 21 or 22 ppts of bromine, like about year 2000 in the stratosphere. Most of them were emitted from anthropogenic sources, but because of their long lifetime, they were unreactive in the troposphere. They don't affect the tropospheric atmospheric chemistry, but they could only account for 15 to 17 ppts of total bromine in the stratosphere. So if we consider the natural emissions arising mostly from the ocean of these volatile, very short-lived compounds, then a fraction of these species will certainly go unreactive into the stratosphere and will photodecompose once it crosses the tropopause. But then another fraction of it will chemically react either by absorbing visible or ultraviolet radiation and also reacting with OH, and resulting in a lot of inorganic products, which can have a very complex atmospheric chemistry in the troposphere, including gas phase and heterogeneous phase recycling. So the total amount of source gas injection in blue and product gas injection in red accounts for the difference that we need to have to get the total stratospheric bromine that was observed from balloons and aircraft. So then whenever we talk about these volatile short-lived halogen emissions, then we need always to consider the difference between source gases and product gases. So source gases from the point of view of the stratosphere can directly be transported and reactive until they start to play some chemistry in the stratosphere. But for the product gases, then these species photodecompose and results in different bromine, iodine, or chlorine atoms that can react with NO2, with ozone, and with different species, and participates in many different chemical processes, like sea salt recycling. These inorganic species also are very important to evaluate the washout efficiency. And also there are some heterogeneous recycling processes occurring either on tropospheric aerosols or ice crystals. So what we did was to develop a complex halogen chemistry scheme and implemented that in a chemistry climate model where we recognize the contribution from source gases and product gases and how was the reactive transport of these species. So this is the community Earth system model, and the CAM-CHEM is the community atmospheric model in chemistry. This is the atmospheric component of CSM. And we develop an emission inventory of many halocarbon emissions. We have a latitudinal and seasonal dependence on chlorophyll, which was in the Ordonez, which is usually called the Ordonez inventory. Based on this, we focus, or I will focus now on showing how the total injection of these halogens to the stratosphere changes between the tropical mean and some hotspots, which can occur, for example, on the Western Pacific. So this was a campaign, the contrast and the Atrax campaign that were performed over WAM in year 2014. This plot shows, was something that was really done a few months after the campaign was performed with the preliminary results. So in blue, we have the bromoform, and in light blue, the dibromomethane abundance. The symbols are from observations, and the lines are from our model configuration, the starting configuration that we have before the campaign was performed. And what we saw is that the total values, the total amount of these inorganic compounds reaching the lower part of the tropical tropopause layer was about 5 ppts, but that a fraction of it, for example, only a small fraction of these source gases reaching the lower TTL were being photodecomposed before crossing the tropopause and being injected to the stratosphere. This was later published by Wells et al. in JGR. And what we also found was that the total injection of source gases occurring in this hotspot in the Western Pacific was larger than the recommended value suggested by WMO for bromine stratospheric injection. So in another work, we also compare how large were these source gas injection and product gas injection of bromine compounds to the stratosphere. So in this figure, the filled symbol are the source gases which are decreasing as we move up in the atmosphere, and the empty symbol are the product gases that are increasing because of the composition of source gases. So what we found out is that this the total amount of product gases strongly depends on the representation, on the treatment, the chemical treatment of these short-lived species, that a surrogate treatment that was originally suggested to be capable of the surrogate treatment was supposed to be capable of properly represent the atmospheric impact of these short-lived species in the upper troposphere. And we show that this was not the case, that we certainly need to have an explicit representation of all short-lived halocarbons to properly reproduce observations in the upper TTL. And this is now considered or taken in the last WMO report, where we see that the total injection of short-lived species of short-lived bromine to the stratosphere, more than half of it, like 2.8 ppts of bromine, is already in the inorganic bromine form, and only 2.1 ppts are in the organic form. So using this, we move into understanding or evaluating the impact on stratospheric ozone. So we performed simulations where we added a constant value of 5 ppts to the injection of the total bromine into the stratosphere, and we found out that the ozone evolution, if we computed the total ozone over the Antarctic polar cap, was better represented if we consider the Shirley bromine species. There was an enhanced decrease by about 15,000 units in the total ozone depletion within the Antarctic polar cap. And most interestingly, what we found is that not only the depth of the ozone hole, but also the size of the ozone hole when these Shirley bromine species were considered was higher. We estimated the enlargement of the ozone hole area by about 5 million kilometers, which is equivalent to the chemical healing that was estimated by that time due to the phase out of anthropogenic emissions of chlorine and bromine species. So I now move to iodine, which we've been talking more in previous talk. So basically, this is just to have a picture of how this is represented in the model. So all these species that are emitted from the ocean, once they are in the atmosphere, they release iodine atoms. These iodine atoms react mostly with ozone to produce IO. This is the species that can be measured by DOAS technique in the atmosphere. But also, all these species can react with other atmospheric components like nitrogen oxide and hydrogen oxide, and participating and making a complete partitioning between different inorganic species. Of particular interest is the formation of ultrafine particles, which requires the formation of higher order oxides of iodine species, and also these complex heterogeneous reactions that occurs on the aerosol phase. So during the Malaspina campaign and many other, and this figure combines many other observations of iodine oxides over the oceans, it was evident that iodine was widespread all over the world, but the emission of methyl iodide that was supposed to be the most important emission from the ocean was not enough to reproduce this observation. So we needed to have an additional source, which was nicely explained by Ryan in previous talk. So in our model configuration, we have not implemented this new equation, but the old expressions parameterized in the paper from Lucy Carpenter and McDonald's in 2013 and 14. And these expressions compute the total flux of HOI and I2 based on ozone, aqueous iodide, and wind speed. So it represents more or less 75% of the total iodine that was emitted to the atmosphere. So based on these maps of emissions, which clearly show larger emissions on the tropics as compared to this new expression that we just saw in the previous talk, we determined and we could reproduce more or less the observations that we have, and with important differences between two different chemical approaches, which I will come later in a couple of slides. One of them considering the photolysis of these higher iodine oxides, and the other one neglecting these contributions. But what I would like to highlight now is that because of the changing in anthropogenic ozone, iodine emissions from the ocean are driven by ozone. And ozone has increased a lot by anthropogenic pollution, mostly in the northern hemisphere. So the increase in ozone, in anthropogenic ozone, has increased the emission of iodine, but when iodine is emitted to the atmosphere, it destroys ozone. So this system constitutes a natural buffer for ozone pollution in the atmosphere. This was published by Cristina Prados Roman in 2015. And after that, we went with some colleagues at Venice. They were performing some ice core perforations in Greenland. And by comparing with their results, what we found out was that since pre-industrial times, so since 1950, the increase of iodine in the ice core record tripled until present time. So this confirmed that this natural buffer feedback was taking place and that the emissions of iodine were increasing in the last decades. So returning to the photolysis of the high order iodine oxides, we know, by some laboratory experiments performed on solution, that these species can photolyze, and once they photolyze, they release IO or iodine atoms again to the gas phase. So in our model, we evaluated two different chemical schemes, one allowing these higher order oxides to photolyze, and another one neglecting this photolysis. And what we found out was that if we see the concentration on the x-axis and the vertical profile on the y-axis, what we saw is that if we don't allow the photolysis of the IX or Ys, then these species start to concentrate as we move up in the atmosphere, and they become unreactive. So all the iodine that is being released from the ocean is not capable of participating in tropospheric chemistry. But when we allow the photolysis of these species to occur, then we can see that the net total amount of iodine is partitioning between different species, mostly iodine and IO, which are the blue, and the red, which are the species that can be measured in the troposphere. And comparing with some observations from the group of Rainer Fockhammer, we found out that this is the green line here. This is the results considering the photolysis of IX or Y. We found out that the observation of these observations performed in the tropical regions were mostly reproduced if we consider the photolysis of these species. And based on this, we estimated that there was a considerable fraction of iodine that could be injected to the stratosphere. So the assessment of the last stratospheric ozone report by WMO was that the total iodine injection was below 0.1 ppt. And based on this study, we consider that the upper limit could be a little bit higher than that. So this has for sure implications for tropospheric ozone. I don't want to focus on this because I was focusing on the stratosphere. But basically, we can see all this red line. This is the percentage of ozone destruction in the troposphere driven by iodine. So it can be between 6 and 20%, depending on latitude and height, and for sure, depending on considering or not this scheme of EX or Y photolysis. So this is just to highlight that the partitioning of species, this is the ratio between ozone in the present day versus pre-industrial. This is the ratio of HOX, and this is the ratio of nitrogen oxides. And not only the total iodine emissions change it between pre-industrial, but we can see that the chemical partitioning can change by up to 50% from pre-industrial to present day condition because of the anthropogenic pollution. So lastly, to finish my presentation, let's see on the impact of iodine in the stratosphere. So what Koenig et al. Found in another work was that of all of this iodine that was injected to the stratosphere, once they reached close to the tropopause, there was a conversion of iodine in the gas phase to iodine in the particulate phase that was captured by different type of aerosols, and the iodine content on aerosol was increasing with height. And based on this, we recognize that a fraction like 0.21 ppts of total iodine injected to the stratosphere was in the gas phase, and 0.56 ppts was in the particulate phase. And this result in an increase of the total iodine injection to the stratosphere in a range, which is the latest assessment of the LUMO, between 0.3 and 0.8 ppts of total iodine into the stratosphere. So we evaluate the impact of these iodine emissions on Antarctic on the Antarctic ozone hole, and found out that for the case of iodine, the impact on ozone is mostly concentrated in the lower stratosphere, below 14 kilometers, and also results in a better agreement with observations compared with the WACAM simulation that neglected the contribution of iodine. And most interesting, we found that this iodine, including iodine, results in approximately 10 or 12, 11% enhancement in the ozone hole area. So the area of the Antarctic ozone hole is larger whenever you consider these iodine emissions. And also, which is really interesting, is that the starting point and the closing point where these ozone, when the ozone hole crosses the 220 Dobson unit definition, occurs earlier and later. So, and a final feature is that what we found in that study is that even when this is the impact of iodine in red compared with all other halogen families during the Antarctic ozone hole formation in October, which is a minor component. But then during the summer, where the ozone hole is not important, but where ozone in the Antarctic is really important for the absorbing UV radiation reaching the surface, then most of the ozone depletion is controlled by iodine, which is also another highlight of that study. So with that, I have already passed my time, I think. So thank you very much for your attention.
Thank you for a great talk. Do we have any questions?
We can have questions for Rafael, but if you still have questions for the two previous speakers, you can also ask them now. We can break open the discussion a little bit.
You see, go ahead.
Yeah, hi, Rafael. Thanks for a really nice talk. So I was just wondering, for the iodine injection into the stratosphere, is the increase that you've got here, is this just because you've turned on the ice recycling scheme for iodine, or do you have these additional mechanisms like iodate photolysis in particles? So there's a slide you've got on the gas to particle conversion. So I'm just wondering, you know, the additional iodine, I think from Koenig et al., or maybe it was one of the later papers, there was a mechanism, there was also a mechanism that hadn't been in previous models. So maybe just could you talk us through those mechanisms for iodine release that are going on there and the TTL in the stratosphere?
Yeah, sure. So basically in Koenig et al., a parameterization not based in our global model, but mostly on the observations and the vertical trends on gas phase iodine and iodine content in aerosols. It was observed and it was determined parameterization that changed the composition. The total iodine content was kept constant, but most of the gas phase was being trapped in the particle. And once in the lower stratosphere, most of the iodine was already in the particle phase. What we found out was that many of these, the iodine content of this species was iodate. And what we assume in the study by Carlos Cuevas on the impact of this species on the Antarctic ozone hole was that this iodate was photolyzed once in the middle stratosphere, once it reached the stratosphere. So all the particulate iodine that was being injected was converted back into the gas phase. And then we have a total of 0.7 or 0.75 ppts of gas phase iodine in the stratosphere, which was being transported to the Antarctic area. And over there, like equivalent to the all cross clocks and Brock cycles that take place in the stratospheric ozone, we also included and consider some cross IOCs plus clocks cycles that was also occurring in the lower stratosphere.
Okay, thank you. So do you have any comment on the contribution, the potential contribution of iodine to lower stratospheric ozone trends?
Yes, basically, there is we've seen that inorganic iodine emissions have been increased since the beginning of the century because of the increase in anthropogenic pollution. And we also are aware that the largest fraction of iodine being injected to the stratosphere depends on the product gases, on the inorganic iodine, and not on the organic emissions. So any trend in inorganic iodine from the ocean surface will end up somehow reaching the lower stratosphere, and it will enhance the additional depletion of the other halogens. We also have another work by Villamayor et al. That highlights that the cross reactions between halogens from anthropogenic short-lived species and natural short-lived species is really important to understand the ozone trends in the lower stratosphere.
Okay, thank you.
Any other questions for three speakers?
For the part in the ocean, the part where halogens come out of the ocean, and then the part where they go up to the stratosphere.
Seems like we have no more questions.
Yeah, Bill?
Sorry.
Had to unmute. I just had a really quick question for Leanne. In that you mentioned the potential for exchange reactions between halogens onto the organic carbon, and I was wondering if you might be able to kind of tease that away, get some sort of insight by comparing the change in those ratios to the known ratios in the inorganic halogens in seawater. So as the carbon gets older, does the ratio move more towards the inorganic ratio, which would suggest exchange reactions? Does that make any sense to you at all? I was just thinking that if exchange reactions are taking place, the older the DOC, the more time that would have to manifest itself. And so you may start with something that's well away from the natural inorganic ratios, but it may converge towards the seawater ratios as the carbon ages. Have you ever looked at that at all?
No, I haven't, but that's an interesting idea. And after today, I think I have a much greater appreciation for the complexity of iodine. So it will definitely, I have a lot to learn, I think, about, yeah, the exchange between inorganic and organic and the mechanisms involved.
It's quite quiet. I can see on the other hand. If I may, I just have a small question for Rafael. Just to for a better appreciation, you said that in the model you missed like 5 ppt of very short-lived bromine species at some point. How confident are you in the models at that point? Like, what is the uncertainty associated with these? Because they are very small concentrations. Just to get an idea of like what would be the range of uncertainty.
Actually, I mean, in one of the slides that I show of these vertical profiles of species on the tropical regions that was published in WMO, basically the observations are from, I would say, more than 15 to 20 campaigns performed all over the world in many few years. So, for example, for the case of chlorine short-lived species, we know that it has been a trend due to anthropogenic emissions, because most of the chlorine short-lived species are mostly anthropogenic. But then for the case of bromine, we haven't seen any trend since year 2000 till the present, and most of the observations are more or less lies in the same value. So we don't expect neither a positive or negative trend on the emissions of bromine that is too large. So I would say that most of the campaigns, I mean, for sure, these are short-lived species, so they have like hotspots in different places. So it's important the emissions, I mean, hotspots on the emissions, and also the efficiency of the transport. That is important to consider how well they distribute in the vertical profile. But I would say that most of the observations and modeling studies, all of them, they lie on these results, and we are pretty confident that these values are considerable, mostly for the source gases. I would say that there are much larger uncertainties on inorganic bromine content. What's the total amount of BRO and bromine nitrate and HOBr and all the partitioning on the inorganic side species, both for bromine and for iodine, is really the inorganic fraction, which is more uncertain comparing to the organohalogens. Yeah.
We also have a question in the chat now. I assume it's for Rafael, but it doesn't say. Since it's about very short-lived species, is there a relation between decalcification and iodinated very short-lived species in calcifiers?
I don't know what to say about this. I don't have an answer at all. I'm not sure if that question is for me.
Yeah, Anna Veronica, if you're still here, maybe you can comment on your question.
Hello, good morning. Yes, I'm sorry for not directing very clearly the question. Yeah, the question is for Dr. Rafael. I was wondering if there is any relation between high concentrations in these iodinated VSLS and metabolism in very young species, primarily in the larvarian stages, focus on sea urchins, or maybe another species that you might be focusing your investigation. You could comment about that, if there's a chance.
Yeah, sure. I mean, for sure, there are some dependence on the type of organisms that are producing these species from the ocean. That is not really my area of expertise. So what we have in our model is that most of the biologically driven emissions result in halocarbons, in mostly methyl iodide, and for that species, which represents approximately 25% of the total iodine content in the atmosphere, there will be a dependence, which I'm not really aware of exactly which references to point to develop that study. But regarding the inorganic iodine emissions of I2 and HOI, it's mostly an abiotic process that depends on surface iodide content and ozone depositions of the surface. So for that main dominant species that is emitted to the lower atmosphere, I would not expect to depend on calcifiers.
Thank you.
You're welcome.
And maybe, I don't know, Liana, if you have more insight in these, in like a production from biological species.
I mean, I've just mostly read about potentially stress response, either light oxidative, but yeah, it's all new to me too.
Okay, yeah, lots to discover there, I think. So if there's no further questions, I think we can conclude here. Yeah, if you still have a question, don't hesitate to put it in the chat. But in the meantime, I would like to thank again the 3 speakers for these really great talks. I think it gave a very nice oversight of what is going on with halogens in the ocean and up to the stratosphere. I thought it was very stimulating. So thank you, everybody. And I hope you enjoyed this, and yeah, some more nice halogen work will come out of this. Thank you. And thank you very much to Lily, our IPO from SOLAS, for hosting this seminar. And thanks to the IGAC ECSC to help us with organizing this.
Liselotte, may I have some words? I would also like to thank both of the early committees for hosting this seminar. And I think this model of collaborations really helps to build up the connections between the two projects, SOLAS and IGAC. I think at some point during the previous conversations, there were some ideas about how to proceed the collaborations between IGAC and SOLAS, which was led by Christine George, which was IGAC SSC member, and then a SOLAS SSC member, Talib. And there were several topics suggested already. I think maybe the two early career committees could carry on these ideas and topics, and to further the collaborations between IGAC and SOLAS. Yeah, that will be my suggestions and comments and some 2 cents.
Okay, yeah, thanks, Lily. I hope we'll manage that. I think we can. There's a lot of goodwill in there, I think. I think you agree, Simone, right?
Yes, I think we will definitely keep collaborating in the future years.
Yeah, thank you very much.
Yeah. Thank you, everybody. Bye.